Hello, welcome to your Biomolecules Lab. Previous video you should have already watched should have been sort of an introduction to biomolecules and the major types of biomolecules they are. What I'm going to do now is go over really briefly all the components of the lab that I have here and the tests we're going to do. And if you have your lab notebook, you're going to see that one of the things that we're going to be running these biomolecules tests on is a regular food sample, a type of food I brought in. Just so you know, our food sample is going to be French fries that I brought from home. And we are going to run, I have a whole container full of samples to run and a bunch of reagents to run approximately 5 different tests on. So we're going to test for lipids. We have 2 tests to do for lipids. One is called a grease spot test. The other is a solubility test. We're going to run another test called testing for simple sugars with a test called Benedict's test. It's kind of fun. We're going to do a test for complex carbohydrates or starches with iodine. And then we're going to do a test for proteins called the Buret's test, okay? The substances we'll be testing for this lab include some of the substances I have over here. So I'm going to turn the camera around. I realize it's a little, whoa. So you can see down here, I have a bottle of starch. Have glucose, and then I have this container full of reagents and other things. I have Buret, so this is your reagent for the Buret's test. Distilled water, Benedict's, more distilled water, mineral oil, this is iodine, and we also have, I want to say one other thing I think we have that I'm missing. There's all of our little bottles down here. So one of the things that I suggest doing is making a list of all the substances that we have here. I have a paper and a pen. And I'm going to show you how to make a list to keep track of what all these different substances are and what we're doing with each one. Okay? I'm going to switch cameras.